C'est un grand plaisir d'être ici aujourd'hui, et maintenant I'm going to apologize for not giving a lecture in French, although you'll see that I really would love to be able to. It's wonderful to visit this fantastic institute, and congratulations, so many good things have happened since you opened it, and of course it really resonates with the goals of the Institute and the program that I run at Stanford, which is called BioX. And the X is for all the intersections of all these different disciplines in engineering, physics, chemistry. So I think this is the future. So today, well, I want to address with you the question, what is a developmental neuroscientist doing studying Alzheimer's disease? So I hope I will answer that question by the end of my talk. But here's the idea. So the idea is really to engage, if we could only understand the developing brain and underlying mechanisms that allow for tremendous brain plasticity in the young brain, especially during critical periods of development. If we could understand those molecular and cellular mechanisms, could we use that knowledge to reopen these same kind of plasticity in the adult brain in aid of repair or even in aid of cognitive enhancement? Par exemple, pour moi, wouldn't it be nice to be able to speak French without an accent? I mean, that's sort of my biggest goal, I suppose, here. And I'm going to try to answer that question today in terms of giving you one example where it is, in fact, possible to restore more juvenile-like plasticity to the adult brain. So what do I mean by a critical period of development? Well, there are many beautiful examples, including language learning itself. But what I want to talk about today is an example in the mouse brain where we can actually manipulate and study, since I'm not sure mice have learned how to speak French yet, but it probably isn't past them. So I wonder if you've ever thought about the problem of having both eyes work together in the brain. Why is it that we don't see double all the time? Well, what part of the answer is that is shown right here in this wiring diagram. So in the wiring diagram, you can see that the neural circuits that create binocular vision start right here, where neurons in the lateral geniculate nucleus receive their inputs from the output neurons from both eyes, the retinal ganglion cells from both eyes. And these connections are kept separate in different layers of the LGN. But you already see here that the input from the two eyes is being combined at this step. Then you will notice that the neurons in the LGN send their connections to the primary visual cortex. And yet again, those connections from the two eyes are getting closer and closer to each other in the sense that they're interdigitated with each other. Yes, this is very tricky here to use this little changer. So they're interdigitated with each other into right eye, left eye, right eye, left eye, right eye, left eye patches in layer 4. And the basis of this interdigitation then is to establish binocular neurons, eventually here, that get their input from both eyes and subserve our ability to see in depth, to have binocular vision, and to have vision that is holistic between the two eyes, so we do not see double. Now, the wiring itself provides a structural basis for this unified view of the world in the two eyes. But it's not enough. The brain actually also has to learn how to integrate the information from both eyes during a developmental critical period where binocular vision is essential. And that is what I want to talk about next. So first of all, let me just say that this beautiful interdigitation of inputs from right eye, left eye was discovered many years ago by David Hubel and Torsten Wiesel. And one way you can see this anatomically is by injecting, let's say here, the right eye with a radioactive tracer, a white tracer, that is transported across the synapse in the LGN and accumulates in the presynaptic nerve terminals, only representing the eye that was injected. And when you then look down at the cortex and at layer 4 of visual cortex, you see this beautiful pattern of white, black, white, right eye representation, left eye, right eye, left eye. And at this scale, every little white dot here is a labeled presynaptic nerve terminal that receives its input from the injected eye, in this case the right eye. And these are the famous ocular dominance columns that were discovered by Hubel and Wiesel. And you will notice that the visual cortex here is divided up evenly, 50-50, into right eye territories and left eye territories. So this is exactly the basis then for creating the binocular neurons in the visual cortex. Now, when these were first discovered, these ocular dominance columns, everyone thought they had to be hardwired because they're so beautiful, so perfect, so regular, always, a stripe of equal width. By the way, this is about 0.5 millimeter here, the scale, so a right eye plus a left eye stripe would be about 1 millimeter. But Hubel and Wiesel did a very famous and important experiment trying to understand the basis for why it is that children who are born with congenital cataracts are permanently blind in the eye with the cataract, even if the cataract is corrected at later ages, after the age of 6. And they made animal models of cataract simply by sewing the lids of the animal's eye closed, and they discovered that even a brief eye closure, a lid closure of only a few days or to a week, profoundly changed these patterns. In fact, here's an example where one eye was closed for a brief period of time, maybe a few weeks here, during an early developmental period to mimic the childhood cataract, and then the open eye was labeled with the white tracer, and you can see the massive change in the pattern of these ocular dominance stripes. The stripes representing the open eye get wider, and those representing the closed eye get smaller. And that's because this is a kind of a use it or lose it process, and it demonstrates directly that this system of binocular vision actually requires a experience in order to develop properly. So even binocular use gives you the normal pattern, and uneven use results in abnormal representation of the open eye at the expense of the closed eye. And this is a beautiful example of what is known as ocular dominance plasticity. So this system is not hardwired, it's susceptible to experience early on in life. In other words, it's plastic. However, if there's normal vision during early life, and then one eye is closed at later times, there's no effect on the wiring. In other words, once formed, this system is stable. And that demonstrates the concept that there is a critical period of development. In children, actually, it's about 6 years of age. In mice, it's during the first month after birth. But regardless, in all binocular animals, there's a critical period of development when binocular vision is required to establish normal binocular function in the visual cortex. And the other point I want to make is that if vision is not intact during that period, then in fact, there's the eye that is not used is blind, and there's no recovery possible afterwards. And that is known clinically as amblyopia. So a profound loss of vision in the eye that was not used. Okay, so I've introduced the concept of a critical period of ocular dominance plasticity and the need for experience during this period. Now, this was discovered many years ago, and ever since then, many of us and the visual system community has been studying the mechanisms of what actually is going on during these developmental critical periods. And the answer is that there's extensive activity-dependent synapse pruning or strengthening, as I've just showed you. So it's a real use it or lose it process with the connections that are heavily used being strengthened, and the connections that are not used or are used inappropriately being weakened and eliminated. So a number of years ago now, we asked the question of how does activity and use drive this process of synapse strengthening or pruning? And in order to address this question, we thought maybe we could discover genes regulated by activity in the visual system. And if we're lucky enough to discover those activity-dependent genes, we could study whether or not they're actually required for circuit tuning during these developmental critical periods. So to try to do an experiment to make this kind of discovery, we blocked activity in the visual system during development. And then we conducted a screen looking for differential expression of genes in the visual system itself. And we did this experiment a very long time ago. And in fact, we were using a differential display approach to looking at gene regulation because it was even before microarrays. And we discovered a candidate, a very interesting candidate, present in the healthy brain and very much downregulated when activity was blocked in the visual system. And when we actually sequenced this candidate, we were hugely surprised because we discovered that, well, we thought we were in the wrong system, not even the visual system, we were in the immune system. Because what we had discovered is that this particular candidate was identical to a known major histocompatibility class 1 family member, MHC1 family member. Okay, so I will tell you what these genes are, but they're famous in the immune system. They're required for adaptive immunity, for cell-mediated immunity. They're known as the HLA in humans, and in both humans and in mice, this is a very big gene family. And what's interesting is, just a little something to mention, is that in humans, this HLA gene family is located on chromosome 6, in a region of chromosome 6, the MHC class 1 locus, which is the most highly implicated locus in the SNPs there in schizophrenia. I mean, in the Manhattan plots of the GWAS studies. And in the context that I'm going to talk about today of pruning, synapse pruning, this is really worth thinking about, and I'm happy to talk about it later. So when we first made this discovery of MHC class 1 expression in the brain, everyone said, oh yeah, well, it's because the immune cells are in the brain. And that's possible. But in the healthy brain, MHC class 1 is not thought to be present. Yet, what I'm going to tell you is that MHC class 1 mRNA and protein is not only present in the healthy brain, it's expressed in the neurons, it's at the synapses, and it's required for this activity-dependent synaptic pruning process. So this is just a little example of many studies showing that at the mRNA level, this mRNA is present. Different class 1 family members are present, both classical and non-classical for aficionados, expressed not only during development but also in the adult brain, different cortical layers, hippocampus, substantia nigra, hypothalamus, any of your favorite places. And it's highly regulated. The different family members are highly regulated. So now I want to make a long story short. I want to simply tell you that many lines of experimentation, not only in my lab but in other labs, have demonstrated a requirement for certain specific family members in synaptic pruning, both in the visual cortex and also at the synapse between the retinal ganglion cells and the LGN neurons. So this is where we like to do most of our studies. We know these circuits very well. So MHC class 1 proteins are cell surface proteins. They are not thought to signal themselves. They are known to present peptide to the famous T cell receptors in conjunction with a light chain called beta-2 microglobulin, which is required for the stable cell surface expression of, sort of like a chaperone, of most, if not all, MHC class 1 proteins. So in thinking about how MHC class 1 proteins, including these two, D and K, which are in black 6 mice, how they might work in neurons and at synapses, we thought perhaps they're working a bit like they do in the immune system, in the sense that there might even be MHC class 1 receptors, not necessarily the T cell receptor, but more likely receptors that are phylogenetically, evolutionarily much earlier. So you know that in addition to the adaptive immune system, which involves the T cell receptor recombination, huge diversity in the ability to recognize peptides, there's also an older system known as the innate immune system. And there are many, many innate immune receptors that have been studied not as well as the T cell receptor, but that are known to recognize MHC class 1 and bind it and signal. So in order to see whether that might also work in the nervous system, we conducted another screen looking for expression of these innate immune receptors in the brain. So they weren't supposed to be in the healthy brain either, but already we had found stuff not in the brain, so we thought let's look for other things that aren't supposed to be in the brain. And to make another long story short, I want to tell you about one such innate immune receptor, which is called PIR-B, and its role in pruning and plasticity. So, by the way, we found quite a number of interesting innate immune receptors expressed in the brain and in neurons, including other members of Lears and KIRs. So these are killer cell receptors. Lears I'm going to come back to in a minute. What is PIR-B? Paired immunoglobulin-like receptor B. This is the LILRB family in humans. If you're studying rat, be careful. The NCBI nomenclature was wrong until we wrote to correct it in January of this year. PIR-B and also the LILRBs are Ig-like. They're beautiful. They're like adhesion molecules, basically. There are 6 extracellular Ig domains, and then a transmembrane domain, and then signaling domains, which are known as ITIM domains, immunoreceptor tyrosine-based inhibitory motifs. And when these are phosphorylated in the immune system, they're known to recruit SHP phosphatases, and in the immune system, that recruitment is known to antagonize downstream signaling cascades, including adhesion and kinase, MAP kinase cascades. And in the nervous system, those are the very signaling cascades that are required for neural synaptic plasticity. So we got very interested in these in PIR-B. And so the other reason we wanted to study PIR-B is that its expression is very clear and beautiful. So at the protein level, it's expressed in pyramidal cells of the neocortex and in the hippocampus, also in the dentate gyrus. Amazing expression in the nose. I have no idea what it's there for. Cerebellum. So there's specifically, they're beautiful places where PIR-B is expressed. So we were very motivated to study PIR-B in the context of ocular dominance plasticity. Here's the visual cortex right here, our favorite part of the cortex. And so we've generated both conditional and germline PIR-B knockouts, and I'm going to tell you a little bit about some of the phenotypes in these mice lines, but there's much more that I don't have time to talk about. What happens to ocular dominance plasticity in germline knockout mice lacking PIR-B? So let me just remind you again about ocular dominance plasticity. Remember, I told you that during a developmental critical period, if one eye is closed, then the open eye, the representation of the open eye in visual cortex expands. Now, the good news is that in the mouse, there is a visual cortex and a binocular zone, and there's a critical period. The bad news is that the mouse cortex, visual cortex, in relation, let's say here, to the monkey visual cortex, so there's a teeny little region of, a few millimeters in size that represents for the mouse the visual cortex. But the good news is that within that, the representation within the binocular zone of the ipsilateral open eye expands. So you can measure that expansion. So one can simply close one eye during the critical period and just measure the width of the representation of the ipsilateral eye and expanded eye and compare it to the normal width. And when you do that experiment in a wild-type mouse, you get the results shown. So you got all the results here. You get the results shown here. So here is a wild-type mouse. Here's the normal width of the representation of the ipsilateral eye, which is the eye that's open. And here's the expansion, the open eye expansion. We used an immediate early gene instead of transneuronal tracing, in this case, much easier to do. So we expected if we knock out a gene, there should be no ocular dominance plasticity. Very stupid, simple-minded, no gene, no plasticity. Wrong. What we discovered is, in fact, that if anything, there is even more expansion of the open eye during the critical period, which is amazing and was unexpected. But remember, the signaling, even in the immune system, suggested it might be a repressive signaling, so maybe in retrospect. Anyhow, not only that, but another really interesting thing is that, as I told you, in wild-type mice, in the adult, if one eye is closed in the adult, there is no expansion of the representation of the open eye because the critical period has finished. However, this was interesting because there still is a significant expansion of the open eye in adult mice lacking PIR-B. And as I get older myself, I am really interested in this small amount of plasticity that's present in the absence of PIR-B. And, I mean, not to joke, but actually seriously, could this be a pill? Could this be the basis of a pill? So let me talk about this now. So what is actually happening? What are the underlying mechanisms that PIR-B is regulating that allows for this extra plasticity? So first, let me talk to you about the idea that maybe there are changes in rules regulating Hebbian synaptic plasticity in the visual cortex. So we all know about LTP and LTD in the hippocampus, but it's also present in the visual cortex. And very beautiful experiments from Mark Baer's lab have even shown, I think convincingly, that in order for the closed eye to weaken during these critical periods, the synaptic mechanism of long-term synaptic depression has to be present at synapses, particularly layer 4 to 2-3 synapses in visual cortex. Those are the synapses that are building the binocular neurons. And there's a little more debate, but I think many of us feel that the mechanism of long-term synaptic potentiation is required for open eye strengthening, at least in part. So what happens in PIR-B knockout mice? And to make a long story short, what we discovered, George Vidal, who's finishing his PhD in my lab, and Maya Jurisic, who's a postdoc in the lab, who, by the way, is on the job market. So what they discovered is by doing synaptic physiology experiments that LTD is impaired. It's not there at the normal pattern of stimulation. And if anything, LTP is enhanced. So what I mean is then that there's excessive levels of LTP. It's elevated and LTD is missing. Now, that's interesting because we realized that if there's no LTD, how could you prune a synapse away if you can't weaken it? So maybe the synapses are stable and you can't actually prune them. So perhaps we ought to also look at the mechanism of synaptic pruning, because remember I told you that during these critical periods of development there's excessive both strengthening of synapses that are used and pruning of synapses that are not used. So to study synaptic pruning, these days there are very beautiful lines of mice. We can actually cross the PIR-B wild type or knockout mice with lines of mice, like these YFP line here, that labels the layer 5 pyramidal cells. You can also study other pyramidal cells, but here the layer 5 pyramidal cells and their apical dendrites are beautifully labeled. And if you look at high power, you can see the dendrites have these little protuberances. These are the dendritic spines, which are the postsynapse. So we can count these dendritic spines and ask whether or not there is a difference between wild type and knockout in the number of synapses. In other words, is pruning normal? And Maya Jurisic discovered that, in fact, there is a defect in synaptic pruning. At the end of the critical period, when synaptic spine density should be here, it's elevated, and it remains elevated into adulthood. And not only are there more synaptic spines in PRB knockout mice, but these spines are more stable. So if we perform an experiment of in vivo imaging, where we make a little window in the mouse brain and look through the window using a 2-photon microscope at those same dendritic spines I just showed you, but now in real life, and measure their motility over a period of hours, we find that there's less motility, there's more stability in the spines in the PRB knockout mice. Okay, so the results of these experiments suggest that PRB is not only required for plasticity during the critical period, that when it's removed, there is more ocular dominance plasticity that persists into the adult brain. And not only that, but this is manifested at the structural level by the persistence of excessive numbers of dendritic spines. Okay, so when we made this discovery, actually, let me just stop here for 1 minute. Of course, everyone said, oh, that's cool, but that's because the immune system in the PRB mice is mutant. And of course, because the immune system is mutant, it's the immune system that is regulating pruning in the nervous system. And, I'm sort of saying to them, well, that may be true, but, these genes are expressed in the neurons, so why can't the neurons do their own thing? And, why can't they determine their own fate? But there's this dogma. So we use the conditional allele to do a really nice experiment that all developmental neuroscientists or biologists will understand immediately, which is a Mosaic experiment. So the experiment is this. With the conditional allele, we can remove PRB function just from single neurons located in the brain in a sea of wild type. So here's an example of that, where we remove PRB just from a single neuron. Everything around it is wild type. The neurons are wild type. The glia are wild type. We do this using a nice developmental trick where we put in Cree with the GFP to mark the cell very early in development, when only those layer 2-3 neurons in this case are being born. So you only remove at very low density PRB from just a few neurons. And then George recorded mini EPSCs from those neurons to assess, and he also counted spines. And when he counted the spines on the layer 2-3 neurons in this case, he found that spine density again was elevated, both apical and basal. So this experiment extends the results. It says that also the layer 2-3 neurons require PRB function to regulate spine density, and also that they can do it without help from PRB expression in other cells in the body or even the brain. In other words, PRB expression in neurons is sufficient to regulate spine density in that neuron, in those neurons. So I hope you understand that experiment. Doesn't mean that you don't need PRB elsewhere. This is a very nice demonstration of sufficiency. Okay, but then what about the glia? Because there are many lines of experiments, including those from Bambaris's lab, Beth Stevens' lab, that have shown that microglia are involved and required for pruning of synapses. And so I would say to you, yeah, it takes two to tango. The glia have to know, though, which synapses to prune. So we believe that PIRB, probably working with MHC class 1 proteins, are part of the neuronal tagging mechanism that is identifying the synapses based on their patterns of activity. And then the glia are using that information to recognize those synapses and prune them. So it takes two to tango. Okay, so let me just now summarize what I've said. So I've suggested that there's a co-regulation of both synaptic weakening and synaptic pruning by PIRB. And now, this is where, oh, it did work. We could not, I'm sorry, it's so loud, just because there's going to be another one. So we couldn't get them to work before. Anyhow, the light bulbs went off. So 2 light bulbs went off, actually. So I want to tell you about both light bulbs now. First, the first light bulb. Okay, there won't be any more light bulbs for a while. So we realized that these phenotypes in PIRB loss of function mice are just the reverse of what happens in Alzheimer's disease. So here's the connection to Alzheimer's disease, where it's known that there is excessive and maybe even indiscriminate pruning of synapses. I mean, you know, synapses are where your memories are stored in Alzheimer's disease, where in fact LTP is suppressed in the presence of soluble oligomeric forms of beta amyloid, and where beta amyloid is thought to be the bad guy here, the causative. Let me just remind you a little bit about these A beta 42 oligomers to just say that normally the amyloid precursor protein, which by the way is expressed very early in development at high levels throughout life, and about which people know almost nothing about its normal function, which is something that's got us very interested now, and I think at quite a number of other labs. Why is it normally there? In any case, normally there's a very highly regulated process, cleavage process, to produce peptides from the precursor protein. There's a cleavage here that produces this moiety, which is then further, there's then a second cleavage by gamma secretase, which produces this A beta 40 normally in wild type peptide. Now, in human Alzheimer genetic patients, a mutation in APP or a mutation in gamma secretase is known to produce a 42 amino acid peptide rather than this 40 amino acid peptide. And this 42 amino acid peptide is known to oligomerize, to produce, to start with, soluble oligomers of A beta 42, which then ultimately lead to the plaques and the fibrils associated with late-stage Alzheimer's disease and cognitive loss. Now, many neuroscientists, I think these days, believe that the process of cognitive loss begins way before even these plaques and tangles can be detected, probably driven by A beta 42 oligomers, soluble oligomers, and that's what I want to talk about now. So for example, experiments from the Malino lab and from the Selko labs and others have shown that in the presence of A beta 42 oligomers, hippocampal LTP is completely blocked, for example. So if you cut a slice of hippocampus and simply look at the ability to produce long-term synaptic potentiation at the synapse between CA3 and CA1 pyramidal cells in hippocampus, then in the presence of beta amyloid, the soluble form, LTP is completely blocked. So we wondered, what about in the case of PIRB? Is PIRB somehow involved in this process? So let's compare LTP induced in the presence of soluble oligomers of beta amyloid plus or minus PIRB. And so amazingly, what we discovered is that PIRB knockout mice have intact long-term synaptic potentiation at this synapse even in the presence of these oligomers, whereas, like everybody else we found, that A beta 42 oligomers prevents the induction of long-term synaptic potentiation. So this is, you know, in the wild type line. What is going on here? So at this point, I'm now going to make a bunch of long story short, but it occurred to us maybe we should cross the Pier B mice with transgenic models of Alzheimer's disease. So in fact, in collaboration with a wonderful neuropathology fellow who was working in my lab at the time at Harvard, and Brad Hyman, we obtained their mouse model of Alzheimer's. It's a transgenic mouse. It carries the Swedish mutation in APP and a deletion mutation in presenilin-1, gamma secretase. So it has two mutations. And these poor mice actually get plaques and suffer memory loss at about 9 months of age. So what happens to memory mechanisms in mice that lack Pier B? So we tested them on various tests, and we discovered that in fact mice lacking Pier B but carrying the transgene, and by the way having very high levels of beta amyloid at this age, including plaques, are completely protected from memory loss, while their litter mates, hets that just carrying even just one copy of Pier B, have memory loss just as expected. So somehow Pier B is involved in generating this process of memory loss by 9 months of age. And removing it protects the mice. We don't know for how long, but at least up to 9 months of age. And so how does this happen? So Tae-Ho Kim, who was a postdoc in the lab and actually is now, it's kind of wonderful, he went to Novartis. They hired him to work on making drugs, which I hope he's going to make really fast for all of us. He hypothesized that perhaps, remember, Pier B is a receptor. So perhaps Pier B is hijacked, is actually being hijacked by, this is really weird because it's going the wrong way, but I'm giving you my whole talk, so I hope that you're enjoying it. Okay, here we go. Let's get to this picture. Okay, so normally I told you Pier B actually binds MHC class 1, and it's known to do so on its two N-terminal domains called D1, D2. And Tae-Ho hypothesized that maybe the soluble oligomers of A beta 42 might be hijacking the receptor. Remember, loss of function causes a pruning defect. So maybe this is a gain of function causing excessive pruning. So in fact, we measured both on neurons and also in heterologous cells a very nice binding affinity and dissociation constant for the oligomeric form of A beta 42. And if you're interested, all of the gory details are in this paper. You can imagine we're not in the Alzheimer's field, and I can tell you the reviewer required us to do every experiment, but we'd also done a lot of them. So there are many details if you're interested in this story in more detail. However, really showing that there is a binding affinity of the oligomers for Pier B. Now this is not a popular idea, since it's thought that it's really later, it's the plaques that are the causative agent. But as I said, many of the current, even the big pharma Alzheimer's studies, which have been targeted against dissolving the plaques, have not been working well. So it's time to start thinking a little bit out of the box. So here's another way of thinking, that maybe earlier receptors, especially at synapses, might be hijacked. And there's a very nice paper by Leonard Mucke's lab showing that FB2, one of the ephren receptors, known to be at the synapse, is also hijacked, can bind beta amyloid. Okay, so when we first published this, there were a number of news and views, that were also followed the paper. And one of them said, yeah, well, but it's known that beta amyloid is sticky. So it's probably just sticky. So, you know, I mean, I will submit to you, it's very hard to measure a good KD of nanomolar affinity with stuff that's just sticky. But in any case, at that time, Michael Birnbaum, who is a neuroimmunologist, trained in a great lab that works on immune receptors in Chris Garcia's lab. Michael came to do a postdoc in my lab. By the way, he's now an assistant professor of biological engineering at MIT, and I know he's looking for really good students and postdocs to work with him. So we use this technique known as surface plasmon resonance to measure the interaction of A-beta 42 with PIR-B displayed on these surfaces. And we can measure a genuine monomeric interaction. This is basically the gold standard. This is a real biophysical interaction, and it's got us very excited about continuing to look at the direct relationship between PIR-B and beta amyloid, especially monomeric and oligomeric forms of A-beta 42. So stay tuned. We can't do it by ourselves, but this is where these great collaborations between across disciplines in BioX really have, you know, been wonderful. Okay, well, PIR-B is a receptor. So what's going on downstream? And so in order to answer this question, Tejo conducted a mass spec screen. And again, I'm going to make a long story short to say that among the interactors that he discovered some very interesting candidates, including an association with cofilin and with members of the protein phosphatase 2 family, including calcineurin. And what's really interesting about these is that they're involved and required both for regulating the actin cytoskeleton, which is part of the stability of the dendritic spines, not to mention presynaptic nerve terminals, and also is required for long-term synaptic depression. I mean, calcineurin is a critical requirement for LTD. And what Tejo then also found is that there's actually an alteration in levels of phosphocofilin in mice, in the Alzheimer model mice, transgenic mice, carrying a copy of PIR-B, but that is manifested by a decrease in phosphocofilin levels. And that's interesting because a decrease in phosphocofilin is known to activate cofilin. And by the way, that decrease in phosphocofilin is actually rescued in the transgenic mice when you knock PIR-B out, which is cool. So this is consistent with the idea that PIR-B is binding beta amyloid and as downstream signaling somehow involving spine stability is being affected, leading possibly to the pruning or the deconstruction of the synapse. So how is this relevant to anything besides mice? So in human, I told you there is a family, a related family called the LILRB family, leukocyte immunoglobulin-like receptor B. This is a family of 5 related LILRs. 3 of them are very closely related in terms of sequence to PIR-B. And so we looked at these in the context of whether or not they also could bind beta amyloid, the oligomers. And again, I haven't told you this, but we've done an extensive domain analysis looking for binding on these various domains of the IG domains of PIR-B and all these LILRs. And to make another long story short, beta amyloid, the oligomeric forms, only bound to LILRB2 and only to the D1, D2 domains of LILRB2, which, by the way, are highly similar to the D1, D2 domains of PIR-B, which is kind of comforting. Not only that, but at the protein level, LILRB2 is expressed in the human brain, both non-AD and D. I don't think there's a significant difference in expression levels here, but what is significantly different is that in human Alzheimer patients, just like in the mice, there is a significant decrease in the levels of phosphocofilin that we could detect. And we only have 4 AD patients here. It'll be really interesting to look at more of them. But already one can see a related change. Okay, so now I'm going to summarize again and tell you that so far what I've talked about is we think that we've discovered a synaptic system that regulates both pruning, and it kind of co-regulates pruning and plasticity, and that when in the presence of oligomeric forms of beta amyloid, we think this system is in overdrive, driving too much pruning at the expense of strengthening. And in knockouts, at least in the mice, we can protect against Alzheimer's phenotypes, and we can also, in the mouse, stimulate somehow growth of and excessive synapses, so we can block pruning in the mouse. Okay, get ready. Here's where the second light bulb goes off. So I'm going to now summarize, but also give you one more set of experiments. So here's the second light bulb, right? There's a drug in here. So there is a pill in here, right? Because this is a receptor. So we can target that receptor in a number of ways. We could certainly do, big compound screens, which would be cool. But as proof of principle, what can you do to actually use this knowledge to make a proto-drug? Well, what you can do is make a soluble mimic of PRB that includes just the extracellular domains of PRB, right? So you can make a recombinant protein that is a soluble mimic of PRB, and this soluble mimic will act like a sponge and will bind to all endogenous or damaging ligands, including the endogenous ligands and also beta amyloid if it's around. So how can we take advantage of this knowledge to even just test whether this kind of approach might work? We're good at doing things like making, recombinant proteins, so we can make this soluble PRB, S-PRB. We can express it, we can purify it. It's tagged with both a MIC and a His tag. We can follow it wherever we put it. Okay, but it's not going to go across the blood-brain barrier, but in my lab we're also quite good at putting things in mini pumps and mini pumping stuff into the brain, so we can put it right into the visual cortex. And what can we do? Well, one question would be, can we just take normal adult wild-type mice that have had good vision their entire lives, and can we put soluble PRB in their adult brains in the visual cortex and ask whether we can reopen the critical period for ocular dominance plasticity? And since we also know that when we see ocular dominance plasticity, it's accompanied by an increase in spine density, let's just check the dendritic spines also and see whether or not we caused an acute increase in dendritic spine density. So we looked at both things, and amazingly, just about 10 days of infusion of soluble PRB caused an acute increase in dendritic spine density. So this is in the adult cortex. To me, this is amazing. And this is kind of a standard way. I mean, this is a drug already. People use this drug kind of approach to treat macular degeneration. This is like an anti-VEGF receptor kind of drug. So this is, to me, amazing. And moreover, this increase in spine density is accompanied by an increase in functional. This is really crazy because this is actually going forward and I'm pressing the reverse. But anyhow, I think I am anyhow. So it's increased in functional spine density and functional synaptic inputs onto these layer 5 pyramidal cells that we're monitoring. It's also increasing functional synaptic inputs to layer 2-3 pyramidal neurons. So we can make recordings, whole cell recordings from these neurons, and we can monitor mini excitatory synaptic currents, or mini, we can look at their frequency and their amplitude, and we find a very significant increase in mini frequency, which is consistent with the idea that at least some of these spines become functional in some way. And as I said, if we measure open eye expansion in the adult visual cortex of mice that have received soluble peer B, but not a BSA control. So BSA is about the same size, another protein. Then we see actually there's open eye strengthening that has happened there. So to me, this is kind of amazing because it says that we can induce the formation of new spines and ocular dominance plasticity in the adult visual cortex. It's also saying something I think quite important, which is that the adult visual cortex, and presumably the adult neocortex in general, has more plasticity in it than is normally engaged. And by blocking mechanisms that repress this plasticity, it's possible to reopen, or it's not even reopen, but tap into those endogenous mechanisms that are already present. So finally, I just want to ask another question. And the question is it possible to use this as a therapeutic? Remember, I told you that children who are born with a congenital cataract, and if that cataract is not treated early in life during their critical period, even if it's treated later in life and the normal optics of the eye are restored, the children are still blind or have extremely low vision, so they cannot recover, even if the good eye is then patched. You can patch the good eye during the critical period, get recovery, but not afterwards. So can we use this approach of soluble peer B to restore vision in a mouse model of cataract, so in a mouse model of amblyopia? So how would we do that experiment? So the way we would do that experiment is make the mouse model of childhood cataract by closing one eye for long-term monocular deprivation, long-term period of time, throughout and actually even beyond the critical period in wild-type mice, and then open up the eye afterwards, and then sadly the mouse is blind in that eye with no functional recovery, and then wait a little while, a few weeks later, infuse for just 7 days the soluble peer B into the visual cortex, and then assess spine density and visual acuity in the visual cortex about a week later. What happens? So first I have to tell you that when one eye is closed for a really long period of time, then there's actually a loss, a fall in spine density and visual cortex. It's a denervation, it's a lack of use loss. So there's actually synaptic loss and functional loss and structural loss here. But when we infused soluble peer B, following this period, we could actually restore spine density to the level of normally reared wild-type mice, not quite to the level of normally reared wild-type mice that get soluble peer B infusion. So that was the first experiment I showed you. But this is really cool. So we're actually putting back synapses. Can these synapses be captured for function, for restoration of vision? So how would we study that? Well, one way would be to have mice and go test them behaviorally. And we're doing that now, but we thought, gee, before we test them behaviorally, we ought to just check and see if we restored visual function in the very place that we treated. Because if we can't test them, if they don't show anything behaviorally, then we won't know what happened. But let's just go into the visual cortex and see whether or not we restored function from the deprived eye. And it's possible to do that, again, by just checking their visual acuity. So you can have mice looking at a screen, and then you can test their visual responses, so their visual evoked potentials, to really coarse gratings or really fine gratings. They don't see really fine gratings well, but you can work out a visual acuity function for cells in the visual cortex. And here at coarse gratings, there's a really big evoked potential. And here, the fine gratings, it's really small, and then it goes away. So you can actually test their range of visual acuity in the visual cortex. And when one does that experiment in mice in mice that have had long-term monocular deprivation, the white line here, they suffer at least an order of magnitude loss of visual acuity, and it's partially restored by just 1 week of soluble peer B treatment, darn it, why didn't we treat for 2 weeks? Well, we're looking at that now, because perhaps if we had treated for longer, we would have fuller functional recovery as well. So the results of this experiment, I think, sort of a proof of principle that this kind of an approach, manipulating this receptor, and in this specific instance, blocking the receptor function, can actually be used for a kind of a model of functional recovery in sensory deprivation. Now, whether or not it could be used to restore synapses and spine loss in Alzheimer's disease is a huge another question that we're extremely interested in studying now. But let me just say one last thing about this, and that is, I think the lesson also learned here is that inflammation is not just in immune cells, in the immune system. So this was a very nice paper. It was a big expression analysis. So gene tip study of changes in gene expression in human brains from late onset Alzheimer's disease. And there were some very fundamental, actually it was kind of funny for me, because the main title of the paper was Decrease in Synaptic Gene Expression in Late Onset Alzheimer's Disease. But to me, the real message of the paper is increase in synaptic expression of all of our favorite genes, which were ascribed in this paper to the immune system. But many of these genes, including, of course, many of the MHCs and these HLAs, a number of them are the class 1 genes, complement, which is also known to be expressed in neurons. And if you look, there's some LILRs even here. So it's not just the immune system that is involved in this process. It's also the neurons, and many of these molecules are at the synapse. So with that, I just really want to thank you again for the invitation. And Eve, thank you so much for organizing this day, which already has been really fun. I already had a great time this morning. And I want to also thank many, many collaborators. We couldn't have done all these experiments in my own lab ourselves. So in addition to people in my own lab working on these questions, we had fantastic collaborators who are experts in immune receptors. We had clinicians who work on Alzheimer's disease. We had colleagues who work on the visual system of the mouse, colleagues who do in vivo imaging experiments looking at spines and synapses, and colleagues who do also imaging of visual cortex using non-invasive methods from actually Mark Hubenier and Tobias Bonhoeffer, and then a great neuroscience behavior lab in our core facility, which is thank goodness, because we wouldn't know what to do studying mouse behavior. So thank you, everybody.